My name is Kyudong. I'm from Dankook University. First of all, I would like to thank PacBio team who organized the PacBio Prism 2024. And today I'm going to talk about solution to accurate human microbiome research PacBio long-read sequencing. And this slide shows the history of the microbial taxonomy profiling. In past study for the identifying microorganisms, we did 16 ribosome RNA gene sequencing about culturable microorganisms using Sanger sequencer. However, these days, many scientists are doing metagenome profiling using next generation sequencing that can apply 16S ribosome RNA sequencing and shotgun whole genome sequencing and whole metatranscriptome sequencing. It doesn't matter if bacterial culture is not possible, just we need to prep DNA from what you want. This figure shows the hierarchy of biological classification from kingdom to species. As you know, short-read metagenome sequencing of the Illumina platform is targeting only V3 and V4 or V4 in the 16 ribosome RNA gene. So classification of biological taxonomy rank down to the genus level is common. But short sequence reading are not easy to classify microorganism at the species level. However, recently, in order to compensate for this problem, companies such as PacBio and Oxford Nanopore are providing long-read technology platform and equipment that can read the full-length 6S ribosome RNA gene of about 1.5 kilobase. As shown in the graph on the right, the long-read sequencing has higher accuracy for species classification within the same samples than the short-read sequencing method. I would like to introduce our one of researches in 2021. At that time, PacBio long-read sequencing was still expensive. That's why we used the synthetic long-read technology using Loop Genomics. That company was just startup company in US. And we found a company called Loop Genomics in 2019. The company developed the Loop-seq synthetic long-read technology optimized for the metagenomics. I don't have enough time to explain it. Please access the Loop Genomics website. And they use the barcode and transposase enzyme. And then finally, you can get the synthetic full-length 16S ribosome RNA sequencing using Illumina platform. This figure shows a schematic diagram of the 16S metagenome library construction process for comparison between the two platforms, Illumina 16S V3, V4. Illumina 16S V3, V4 is a short-read amplicon library targeting the V3, V4 region inside the 16S ribosome RNA gene. However, Loop Genomics is a synthetic long-read library that includes all variable region from V1 to V9. After short-read sequencing, Loop-seq 16S full-length data is formed by recombining unique molecular barcode short-reads. This figure shows how clearly the taxonomy feature ID was classified among bacterial species generated by V3, V4, and Loop-seq platform. As you can see, 98% of the species are unclear taxonomy in V3, V4. However, Loop-seq is about 34% unclear taxonomy, and the rest have clearly identified intestinal bacterial species. We confirmed that Loop-seq technology is good enough to classify microorganisms at the species level in metagenome research compared to the short-read method. However, it is in sufficient technical service in South Korea. Additionally, the cost was still expensive, and also the preparing the library construction is so complicated, and some of the experiment is not reproducible. Therefore, we turned our attention to PacBio's long-read platform. And PacBio is the most valuable, well-validated global long-read sequencing platform. Recently, continuous technical improvement have resolved the most critical accuracy issue of the long-read method. the cost per sample has decreased for its application in metagenomic. Finally, we were able to contact the PacBio Korea, and through collaboration with the company focused on microbiome research. Finally, we established the Skin Microbiome Center with one of cosmetic company, Cosmex. And through an MOU between PacBio, Dankook University, and Cosmex in 2023, we are operating the Skin Microbiome Center by sharing systematic goals. Additionally, in 2024, we established the DKU Cosmex Innobeauty Center, and I received the $2 million in research funding from the Cosmex. That's why we can purchase the Sequel 2E. Yeah. Through the operation of Sequel 2E, we are mainly conducting the full-length 6NES metagenome analysis. In addition, we are doing the whole genome sequencing analysis of microorganisms and RNA sequencing with PacBio's IsoSeq, and targeted sequencing for studying structural variation of BRCA1-2 genes from breast cancer patient tissue samples. This is PacBio SmartBell sequencing workflow for the metagenomic. And the success of data production and analysis for full-length 6NES library depends on how well the 6NES amplicon is completed. This process involved amplifying metagenomic DNA samples using 27 forward and 1492 reverse primer, followed by end repair and ligation with SmartBell adapter. At this stage, PacBio SmartBell system allows us to 192 sample to be indexed. But in our case, we usually run one single flow cell around 120 sample. When we are studying skin microbiome, DNA obtained from skin is very low concentration of DNA. As you can see, the concentration looks okay on the nanodrop, but more accurate quantification with the qubit revealed that the template concentration for PCR application was critically low. Consequently, when we doing the 6NES PCR following PacBio recommended protocol, as shown in the gel picture left side below, some of samples were not amplified. Therefore, we for the failed PCR sample, we either increase the volume of the template, or we increase the PCR cycle up to 29 cycles. And then finally, we got some of samples get the amplification, and then we pulled to put the Sequel 2. And the dumbbell structure of a SmartBell template changes into a circular form during sequencing. It allowed the polymerase to continuously read the sequence until enzyme activity stops. As a result, by comparing the rapidly read identical sequence, a hi-fi read with 99.9% accuracy can be generated. The sequence data are then transferred to a SmartLink server. The hi-fi read are demultiplexed using the SmartLink server integrated within Sequel 2E system. The multiplex read data are then transferred to our own server where we custom built a microbiome analysis pipeline. From the demultiplexed read, we only retain those with a Q score of 20 or higher. Next, it trim adapters, primers from the reads. The result data are then processed in QIIME2, whether the DADA2 package handled the denoising and clustering. And then after that, we use the Silva database. And also, our samples are from sometimes plant and human tissue. That's why we filtered out the mitochondria and chloroplast sequence. And after that, we collaborate with one of our company, Human Biome. They build the advanced analysis, and then we can see the visualized some output using their platform pipeline. And together with Cosmex, we built a skin microbiome data set of 1,000 Korean individuals using MySeq 1 year ago. At the time, there were 3 groups, young and aging and older. And we analyzed 94 samples from the aging group, age 35 to 49, using PacBio for 6-nest full-length metagenome sequencing in 2024, this year. Here, I would like to discuss this result. And as a result, we compared the data yield between the 2 methods when considering only the reads that passed the quality check and were usable for analysis. We found that the V3, V4 method produced a greater number of reads compared to full-length 6-nest method. This is an expected result because Illumina provide about 500 base pair of read, but PacBio provide 1,600 base pair of information for each read. However, the percentage of reads remain after filtering process is significantly higher because the Illumina just left 44%, but PacBio left 67%. And secondly, we examined the alpha diversity analysis result between the 2 methods. The result for Shannon index, Simpson index, and PLO evenness, which indicate the diversity, concentration, and evenness of the microbial species within the samples, show no significant difference between 2 platforms. However, there was significant difference in value representing the number of microbial species or richness, such as observed features and the SHA-1 index. That means PacBio can identify more species than Illumina V3, V4 technique. This slide shows the taxonomy classification top 10 phylum and top 15 genus and top 15 species. The relative abundance between the 2 methods did not show significant difference in composition from the phylum to species level. As expected from an analysis of skin microbiome, both methods demonstrate dominance of the genera such as Cutibacterium, Streptococcus, Staphylococcus, and Rosia. At the species level, we confirmed the dominance of typical skin commensals, including Cutibacterium acne and Staphylococcus epidermis. However, when we examine the species level in more detail, we found that the V3, V4 method identified a total of 892 species, but 478 of these were unidentified species. In contrast, the full-length 6-nest method identified 862 species, of which only 31 species were unidentified species. This indicates that the V3, V4 method has a limitation in accurately identifying species. Therefore, the high number value of observed features and SHA-1 index in the full-length 6-nest method seems to be due to more accurate species classification from the PacBio. Additionally, the V3, V4 method could not distinguish whether the genus Staphylococcus was Staphylococcus aureus or Staphylococcus epidermis at the specific level. However, with the full-length 6-nest method, we determined that most of the Staphylococcus species are Staphylococcus epidermis is 10% and Staphylococcus aureus 2%. This is real ATGC sequencing data. As you can see, for the Illumina MySeq data, the difference in the sequence in the V3 start region alone make it difficult to accurately distinguish between the Staphylococcus aureus and Staphylococcus epidermis. However, for the full-length 6-nest sequence, we were able to achieve accurate classification by considering the sequence difference in V3 region. shown in the previous slide, and additional V1 and V5 region. Therefore, only full-length 6-nest metagenome sequencing allow for species-level classification of closely related species as demonstrated in the current result. Based on the advantage of the full-length 6-nest method, we are now able to provide researchers with a corona chart that allows them to visualize the taxonomy composition of each sample to the species level. Everyone used this tool, web-based Galaxy or server-based pipeline, but our lab used server-based corona. And this is my summary slide. Actually, our university is not that big, and then purchasing the NGS is not easy. That's why we are starting the industry academic corporation approach can simply the process of arranging expensive equipment. And therefore, we are able to quickly construct the PacBio SQL 2E platform and microbiome analysis pipeline with COSMAX, MDXK, PacBio Korea, and HumanBiome. And taxonomy identification through metagenome requires the use of full-length 6-nest ribosome RNA sequencing, and among existing next-generation sequencing technology, PacBio is the best. As a result, we had the opportunity to prepare PacBio 6-nest library of various types: skin, fecal, salivary, animal tissue, and plant. As these experiments accumulate, we are accumulating solution and know-how for sample types that are difficult to prepare for PacBio 6-nest library using formal protocols. And this is my lab members, and we work together and hard. And then, yeah, our lab is 29 researchers in my lab. And I would like to thank COSMAX and PacBio and Korean company MDXK, HumanBiome, and Teragen Bio. And thanks for your attention, and question will be later. Yeah, thank you.